Good morning to all and welcome. This is the technical session number 6 of the first international congress organized by FibEnTech. My name is António Mendes. I'm going to be the chair of the current session, which is dedicated to a very important subject nowadays, the environment. In this section, we will have 4 works covering different topics, all of them within the same main subject. In particular, the first 2 that you will see will be presented by the same person, Professora Flora Silva from the Polytechnic Institute of Bragança. Then the next work of this session will be presented by Professora Flávia Matias from the Polytechnic Institute of Beja. And finally, the 4th and the last work of this session will be presented by Professora Isolina Gonçalves from the University of Beira Interior. They are all in line, and greetings to all of them, all the 3. I would also like to remind the viewers that are watching that if you have questions during this session, please write them on the streaming platform that you are using, or send them to my email address that appears on the screen. And also, please don't forget to include the name of the author that you are addressing. Okay, that's about it. And now, without any further delay, let's get started with the video presentations that were prepared by the authors, and we will start with Professora Flora Silva.
Hello everyone. I will present a paper entitled Water Reuse in a Municipal Sports Center. Water scarcity is one of the leading social and environmental problems faced by society. Consequently, there is a need to preserve water and search for alternative sources. The reuse of grey water flow originated from showers, hand basins, laundry, dishwashing, can be one of the solutions to achieve one of the major goals of urban sustainability, the water and energy efficiency. It has been estimated that about 41 to 91% of the domestic water use turns into grey water. In Europe, is produced around 35 to 150 liters per person day of grey water. Grey water from laundry and kitchen are considered to have a high pollutant load, whereas the water from showers and hand basins has a low pollutant load. Thus, the lead can be reused for flushing cisterns or gardening after simple treatments. Example, filtration, sedimentation, and disinfection, since the water used for the mentioned purposes does not require potable quality water standards. In sports centers, around 72% of the water is used in showers and hand basins. Therefore, it is of paramount importance to implement management measures to promote the reuse of this water in these large area facilities where water consumption is usually high. If these have simultaneously swimming pools, the pool filters rising also generate large amount of water, which can, in turn, be reused. Indeed, this process is required to remove contaminants accumulated during the pool filtration process. The filters are cleaned by flushing them with water in the reverse direction to normal flow, backwashing. The resulting backwash water is characterized by high contents of suspension and dissolved substances, and depending on the future application, require differentiated treatment before being reused. The Bragança Municipal Sports Center has the following facilities: an indoor football court, Building A, 2 indoor swimming pools, Building B, and the local football stadium, C. In the present approach, were evaluated 2 potential measures for increasing water use efficiency in the building: the reuse of water originated from showers in the flushing toilets in Building A, and the reuse of water from swimming pools filters cleaning for irrigation. According to the information provided by the municipality, the average number of users per year in Building A was 36,873, 128 users per day from 2016 to 2018. In the present approach, because of the COVID pandemic, it was impossible to measure in situ the different water uses. Therefore, these were estimated based on audits carried out by ANKIP for water use in sports centers. The acquisition of a commercial recycler is suggested to minimize the gap between the inflow of greywater and its consumption. The amount of water necessary to backwash the swimming pool sand filters, as shown in Table 1, was quantified by registering the values of the water flow meter immediately before and after the backwashing process. Swimming pool filters are rinsed every 2 days for 5 minutes. After the backwashed water is discharged into the wastewater drainage system. To evaluate the quality of backwashed water and the possibility of its reuse for irrigating the lawn of the local football stadium, samples from filter washings were collected in the first and the third minute after the cleaning was started. In the lab were submitted to different sedimentation periods, 0, 2, and 15 hours. Afterwards, samples were analyzed for total suspended solids, pH, turbidity, and total coliforms, fecal coliforms, and streptococci colonies were enumerated. The average annual water consumption in Building A is around 3,500.5 cubic meters. The estimated annual water consumption for showers is 1,785.26 cubic meters, 51% of the total water consumption. Therefore, the greywater generated by showers can be reused in the flushing cisterns, whose predicted consumption is about 840.12 cubic meters per year. The daily consumption for the flushing cisterns is around 2.92 cubic meters per day, and the daily greywater produced by showers is 6.2 cubic meters per day. Therefore, it is recommended the acquisition of water recycler dimensioned for the observed water flux to minimize the gap between the inflow of greywater and its consumption. Indeed, raw and treated greywater should only be stored temporarily in a tank for less than 24 hours to avoid bad odors and the good conditions for pathogens proliferation. To reduce turbidity and total suspended solids to admissible values. The predicted water consumption reduction can reach 24%, and the annual water bill cutback will be €4,594.22. The average daily water consumption for swimming pool filter cleaning was 15.7 cubic meters competition pool and 7.7 cubic meters learning pool. Therefore, the estimated annual water consumption for filter washing were 2,818.8 cubic meters and 1,378.8 cubic meters, respectively, totaling 4,197.6 cubic meters. The highest values for total suspended solids and turbidity showed higher values in the samples collected during the first minute of the filter backwashing, first flux. In those collected in the third minute, a reduction between 10 and 20% occurred in the studied parameters. Concerning total suspended solids, the 2 and the 15-hour sedimentation reduce the amounts of suspended solids by 60 and 80% respectively. Turbidity showed a similar trend. Nevertheless, total suspended solids and turbidity values exceeded the permissible values, except those submitted to 15-hour sedimentation. pH values were always in the admissible range. Total coliforms, fecal coliforms, and fecal streptococci were not detected. The regular swimming pool filters backwashing is unavoidable. Nevertheless, this process generates large amounts of water, usually discharged into the sewage, contributing to the water and energy use inefficiencies. Therefore, it is crucial to promote the sustainable management of filter washings. The preliminary results showed that this water could not be directly reused because it does not fulfill the regulation requisites. However, the sedimentation process seemed to effectively reduce these parameters to permissible values, allowing the reuse of this water for irrigation or flushing toilet system. Therefore, considering these results, it is plausible to assume that the management of filter washings is possible after installing a relatively simple system for their treatment, such as a settler or a setting tank. Furthermore, the annual amount of water necessary for irrigating the lawn of the local football stadium, located in the surrounding of the swimming pool complex, is around 7,200 cubic meters. Filter backwashing could be fully reused in the mentioned purpose. To conclude, in a scenario of increasing water demand and scarcity, implementing measures leading to greywater reuse when quality drinking water is not required for water supply is paramount. The present study showed that it's possible to promote a substantial increase in water use efficiency in the municipal sports center using water reuse measures, which contribute to the financial burden reduction and increasing environmental benefits. Indeed, sports centers have substantial operational costs due to extensive water consumption, indirectly implying significant energy consumption levels. Herein, the evaluation of the energy consumption under the scope of water energy nexus was not evaluated. Consequently, the earnings for implementing the suggested measures are monetarily and environmentally much higher than the estimates obtained herein. In short, the current approach indicates that the reuse of greywater is not only feasible but can lead to a significant reduction of water consumption in the municipal sports centers, reducing the environmental impacts associated with urban water demand. Thanks for your attention. Hello everyone, my name is Flora Silva and I will present a paper entitled Influence of Hydraulic Conditions on Reclaimed Water Polishing Using Soil and Sand Columns. Adaptation and mitigation of climate change through water management are essential for sustainable development. Therefore, it is increasingly necessary to consider unconventional water resources in future planning. Reclaimed water is a reliable alternative to conventional water resources for many uses. Indeed, there are several successful cases of water reuse worldwide, including the artificial recharge of aquifers with treated wastewater. The complementary treatment of secondary effluents through the soil aquifer treatment, before its inclusion in the aquifers, has been gained increased attention in several countries around the world, with studies pointing to the advantage of its application. However, if the soil does not present favorable conditions for infiltration of treated wastewater, the residual loads of that water, characterized by an organic matter of difficult biodegradability, example, hydrocarbons and pesticides, forms of nitrogen, ammonium, nitrite and nitrate, and phosphorus, orthophosphates and polyphosphates, heavy metals and pathogenic microorganisms can be a disadvantage for groundwater quality. Thus, the purpose of this research was to evaluate the influence of the hydraulic conditions on the removal of organic matter and nutrients, nitrogen and phosphorus forms, using laboratory columns filled with soil, the fine fraction of the soil and river sand, and verify the main mechanisms responsible for removing these pollutants. A granitic residual soil was collected at the Quinta Gonçalo Martins, Guarda, Portugal. A component less than 0.075 millimeters fine soil was extracted from the soil, as this is where the most reactive soil fractions are found, namely silt and clay. In addition, river sand was also used. To understand the importance of soil properties in the removal of pollutants, some physical, chemical, and mineralogical properties was determined. Soil granulometric analysis, fine soil analysis, specific surface, density of solid particles and porosity, qualitative mineralogical composition of the soil, morphological and microstructural analysis, chemical composition, analysis in oxides, organic matter, and soil pH. Experiments were set up in 3 acrylic circular columns with vertical and downfall flux with a height of 42 centimeters and 14 centimeters in diameter, and a synthetic effluent. Water samples were collected at column top P1, P2, and column bottom. The 3 columns were filled with soil, fine soil, and river sand. The filling media had a height of 25 centimeters. The first column had 20 centimeters of soil and 5 centimeters of sand at the bottom. The second column had 20 centimeters of fine soil and 5 centimeters of sand at the bottom. The third column had 25 centimeters of sand. A synthetic effluent was used with loads until 150 milligrams per liter of organic matter, 30 milligrams per liter of ammonia, 10 milligrams per liter of nitrate, and 10 milligrams per liter of phosphates, which are typical of urban effluents from secondary treatment. Experiments were run in 2 phases. In the first phase, the columns were operated in a discontinuous mode, including filling with the feed solution, reaction for 3.5 days, totaling 35 days, 10 cycles, drainage, and sampling. In the second phase, the columns were operated in a continuous mode, also for 35 days, with a feeding system composed by peristaltic pump for pumping the effluent to the column's inlet, with a flow rate of 0.36 liters per day and a hydraulic load of 2.34 centimeters per day. At the beginning and end of each cycle, in first phase, water samples were collected at the influent, P1, P2, and effluent for measuring pH, temperature, organic matter, ammonia, nitrate, and phosphate. For the second phase, water samples were collected in the same points each 3.5 days for determining the same parameters. The soil used in the study has a low clay fraction, particle size less than 2 microns, of approximately 5%, which means that most of the fines are essentially silty. To prevent soil clogging and ensure wastewater treatment, the soil should not contain more than 10% of clay. Figures 3a to 3c show the morphology of soil, fine soil, and sand particles. Table 1 presents the physico-chemical characteristics of the 3 materials most relevant to the study. For fine soil, the permeability is low, but suitable for controlled infiltration of treated effluents. It essentially contains silica and alumina, with lower levels of iron and potassium. The organic matter content is very low and the soil is very acidic. The mineralogical composition consists of quartz, muscovite, halite, kaolinite, and smithite, identified in the fraction below 2 microns. Kaolinite constitutes about 60% of the clay mineral present in the soil. This soil has a specific surface area suitable for the development of biofilm capable for removing pollutants and pathogens, and the clay-colloidal complex of this soil also has reactive properties that allow the removal of pollutants by sorption mechanisms. Table 2 and Table 3 show the variation of pH, temperature, organic matter, ammonium, nitrate, and phosphate for the column top and the column bottom sampling points in first and second phases. Also show a drop in pH from column top to column bottom or column base. The average temperature remains practically constant throughout all cycles. The best removal rates were obtained for fine soil, and therefore the analysis will only be focused on the fine soil. The removal efficiencies of organic matter, ammonium, nitrates, and phosphate were high in both discontinuous and continuous columns experiments using soil and fine soil. In the column filled with fine soil operated in discontinuous mode, there were observed removal efficiencies of 80.78% for organic matter, 96.55% for ammonia, 96.81% for nitrate, and 98.73% for phosphate. The removal of these pollutants from the sand was relatively low. In both experiments, carbonaceous removal has occurred either in the presence of oxygen closer to the surface of the column by aerobic removal mechanisms such as aerobic respiration and nitrification, or in the absence of oxygen in anaerobic removal mechanisms such as anaerobic respiration, denitrification, and fermentation process. In the ammonia removal, the nitrification was present. Therefore, ammonia oxidation may have occurred closer to the surface, where oxygen is able to dissolve in the water mass by diffusion. In both phases, the removal of nitrate was very significant, which have occurred through denitrification and the lower layers where oxygen was low. Nitrates were present through the feeding solution and also produces due to the ammonia oxidation, nitrification. The phosphate removal has occurred by biosorption. It is adsorbed on the polymers that make up the biofilm, and by complexation and precipitation at low pH, usual complex with aluminum and iron, precipitating in the form of phosphates. To conclude, the soil and its fine fraction have reactive properties that allow a good capability for removing organic matter, ammonia, and nitrate by biodegradation mechanisms and phosphate by sorption mechanisms, either in discontinuous or continuous mode experiments, allowing it to act as a barrier to the contamination of groundwater during infiltration with reclaimed water. However, fine soil has shown to be a better media for residual pollutant removal in soil. Thanks for your attention.
Hello, I am Flávia Silva, teacher at the School of Agriculture in Ipabeja, Portugal, and I'm going to present the work of our research group coordinated by Professor Adelaide Almeida, together with my colleague Jorge Tavares and the others authors. Our theme is removal of emergent pollutants by vertical constructed wetlands with Vetiveria zizanioides. Case stood for caffeine. Among the pharmaceuticals, caffeine has been widely studied in wastewater treatment system as an alkaloid, a physical active substance, and legal is one of the most consumed in the world, being present in different drinks such as Coke, soft drinks, coffee, tea, and energy drinks. Most pharmaceutical products are not completely metabolized after human ingestion, resulting in the metabolic excretion and other similar compounds that reach wastewater treatment plants. Wetlands are not sized for the specific purpose of removing this type of compounds. Due to its high solution, 21.6 grams per liter, and low volatility, caffeine is quite persistent in water masses. However, it is necessary to understand what are the main mechanisms involved in caffeine removal. The effect of increasing concentrations on plant, biomass, and system performance. Constructed wetland is very efficient from the point of view that increases the ability to remove various types of pollutants, including caffeine. Studies in vertical systems showed that caffeine was partly accumulated by plants. From these positive points, the objective of this work was evaluate the ability to remove caffeine in constructed wetlands under different concentrations of the caffeine. The experimental work was carried out in a well-established 8-year-old pilot-scale vertical flow constructed wetlands located at the Agrarian College in Beja. Was the plant used is Vetiveria zizanioides, the filter material in the light expanded clay aggregates, caffeine concentration at two different concentrations, 0.75 microliters and 1.5 microliters. 1 month for each caffeine concentration was studied. Each new trials was initiated by increasing the caffeine concentration. Every day, samples were collected and inlet and outlet of the VFC double. Vetiveria zizanioides was used and ADPLCMS for analysis to determine the concentration of caffeine in the effluents and effluents as well as in plant biomass after leaf extraction. Plant biomass growth was monitored in both beds on a weekly. Characterization and concentration was made, and chlorophyll, E and B, carotenoids, nutrients, and metals. Results. The biomass growth was not negatively influenced by the presence of caffeine in the influent. There was an increase in the growth of biomass during the second trio. Caffeine concentration 1.5 more or less milligram per liter. The level of chlorophyll A and carotenoids showed an increased tendency due to the feeding of plants with caffeine. However, the chlorophyll B level showed a very strong tendency to decrease during the trios. Sodium and potassium levels showed an increased trend due to increased caffeine level in the influent of the VFC doubles. It was deduced that TSS concentration remained constant over the test and did not occur precipitation of salts during the experiment. The potential redox measured in the inlet and outlet of the VFC levels was always positive. There was not significant difference when compared to the average values obtained for the influent and effluent. The dissolved oxygen of the effluent increased in both trios, possibly due to the capacity of the plants or the type of the flow that allowed the replacement of oxygen consumed by the microbial present in the beds. The outlet pH decreased without statistical significance of both trios. Regarding efficient and effective monitoring of beds, it was obtained considering the difference and increasing concentrations of caffeine, an average removal of 93% and 87%, respectively. These results were similar to obtained with zinc, that obtained remove efficient in hydroponic conditions until 99%. After 21 days of retention times, but the retention times in our system was very low. Oliveira studied the removal of caffeine in a pilot VFC double with an ADRT of 7 days and arrived removal efficient of 97%. Conclusion: the results obtained showed a removal efficient of up to 93% of caffeine using vertical flow constructed wetlands with Vetiveria zizanioides, suggesting a simulation of 10 milligrams by plants. However, given the high caffeine removed content, this was not the main removal mechanisms, pointed to the possible formation of metabolites. Removal by microorganisms, size caffeine is considered a readily biodegradable pollutant. This is reference. Thank you for attention.
Good morning to the audience. I would like to thank you to the organizers for giving me the opportunity to take part in the first FibEnTech International Conference. I'm going to talk about grape pomace as a renewable source of energy, degradation efficiency, and biogas yield. In this slide, a summary of the work is described. The schematic representation of the research work is given in the present figure. The grape pomace is going to be used as substrate of anaerobic microorganisms to produce biogas. The biogas contains methane. After purification, it can be used for several purposes. namely heating and transport fuel, electricity, advanced fuels, and biomaterials production. It also contributes to the climate neutrality. Thus, it is very important to estimate the amount of grape pomace produced from winemaking industries, as well as the amount of energy that can be obtained. As can be seen, Portugal accounted for annual production of 6.5 million hectoliters in this period, being approximately 20 to 25% of grape pomace, which contains skins and pulp, seeds, stems, and stalks. So, grape pomace as a raw material could help to accomplish the forecast of 25% of all European biogas. The objective of the present work is the evaluation of the biogas yield and degradation efficiency of red grape pomace in an aerobic mesophilic regime, particularly the influence of the particle size, initial mass load, and soluble carbon source addition. In materials and methods section, some information is given about the experimental conditions and the main parameters monitored in the systems. The layout of the experimental procedure is shown in this figure. The grape pomace is going to be used as substrate by anaerobic microorganisms to produce biogas. Before fermentation, it was pre-treated by drying, grinding, and sieving. Also, a mineral medium, with or without carbon source, was supplemented to the anaerobic reactors. Results indicate that a significant fraction of organic matter is readily available to be used as substrate, and these results were shown in previous work, and also that an increase in the initial GP mass load by a factor of 2 may lead to an increase in the biogas production up to 3 to 4-fold. The biogas yield up to 144 liters per kilo of volatile solids aids was obtained. However, this value is much lower than that considered the theoretical estimated one, which is around 800 liters per kilo of volatile solids removed. The ICOD removal and phenol removal corroborate the degradation efficiency of solubilized substrates leached from GP. In figures 2 and 3, the results for biogas yield are shown. We see that the biogas yield increases with increasing the initial mass load and decreasing the particle size. In figure 3, the values of the biogas yield estimated per kilo of volatile solid removed and per kilo of volatile solid added are shown, and the results are quite different, suggesting that a significant amount of grape pomace is not broken down within the experimental period, remaining inside the bioreactor. Finally, we can conclude that the biogas yield values calculated in terms of soluble CO2 removal suggest that soluble and easily hydrolyzable compounds undergo degradation. However, a significant percentage of grape pomace is not broken down within the experimental period. Thus, a commitment between the type of pretreatment, the mass load, and the particle size appears to be of utmost importance for the improvement of the overall performance of the system. Thank you very much for your attention.
Hello again. We are back in live and we will now open the debate with the authors. with some questions that have been made during the video presentations, which were very fine. I have a couple of them here with me. And the authors, Flávia, Flora, and Professor Isolina, are you ready for the questions?
Yes, I think so.
Yes.
Okay. So I will start with one. The first one for Flora. Okay. This concerns the work number 1. The question is, did you consider grey water reuse for cleaning pavements? And the question has a second part. What are the standards applied to this type of application?
No, to the wash the pavements. Yes, washing pavements. No, only to the flushing cisterns. Not to washing the pavements. In other study, I will is a case to see this measure, but in this study, no.
But the standards applied to this type of application, do you know? Do you have this information?
Yeah, the washing pavements. I don't understand.
The cleaning of the pavements. The person that asked the question is asking about the standards applied. Do you have this information, or it's not a concern for now?
No, I haven't more information about.
Okay, fine.
Okay.
Thank you for the response. Okay. A second question now for Flávia. Okay, Flávia, I have here a question for you. The question is, was the possibility of caffeine being absorbed by the filling material excluded? What do you think?
Good afternoon, everyone. So I ask my colleague Adelaide Almeida will answer this question, please.
Okay.
Sorry,
good morning, Adelaide.
Hello, but I don't understand the question. Sorry, please, can you repeat?
The question is, was the possibility of caffeine being absorbed by the filling material excluded?
By the material from the bed, is the question?
Yes.
I think related to caffeine, I don't do the work about the possibility that be absorbed, but I worked with another compounds like ammonia, and don't was absorbed by the material, the metric of the wetland.
Okay.
Related to caffeine, I don't know. In the future, I can start the study, but nowadays I don't know.
Okay. But still in this work, I was curious about something. It was not specified in the work. What is the problem of finding caffeine in the environment? Is it dangerous?
Yes, of course.
Yes.
Sorry.
What's the problem of having the caffeine, for instance, in the wastewater?
Caffeine is stimulant the nervous system. In the wastewater, caffeine usually is not good removal in the conventional systems. And if caffeine is put in the water body, can go to water supply or water drink water, and is a big problem for the public health.
Okay. So, but your this methodology can also be used, for instance, for other components such as antibiotics. Yes.
Yes, but I only start with an antibiotic, only one, and because it's very difficult for us work with HPLC, MES, MES coupled to HPLC. It's very difficult. I don't have students. I'm work with Flávia alone, and it's very difficult for us.
Okay, thank you. Thank you very much, Adelaide and Flávia. Very interesting.
Thank you.
I have now one other question now for Professor Isolina. The question is, how to distinguish between biogas production yield due to the biodegradation of the grape pomace from those resulted from the consumption of glucose and acetic acid added as alternative sources of carbon, far easier to be metabolized by the anaerobes? What can you say about this?
Yeah, I have a problem with my phones. Are you listening me? Yeah.
Yes, we are listening.
With my microphone. Okay. Relatively to this question, indeed, the idea of this work was to enhance the biodegradation in terms of not only biogas yield, but also of the liquid treated simulated wastewater that was added or supplemented to the grape pomace. Indeed, this work was a co-digestion between solid waste and wastewater, simulating industrial wastewater. And the idea was to supplement some soluble carbon sources, particularly at high grape pomace loads, initial loads, in order to enhance the overall efficiency of the process. And we have chosen glucose because it is a substrate that can improve the activity of all anaerobic communities, namely of hydrolytic acidogenic bacteria and also acetogenic and methanogenic syntrophic group. And also we tried to add only sodium acetate. The sodium acetate was to observe if increasing just the syntrophic group, acetogenic, methanogenic microorganisms, we will get an improvement of the yield. In terms of biogas, because under anaerobic conditions, most of the organic substrates are converted into biogas, into methane. And nowadays, methane is considered a very, very important source of bioenergy due to several applications in terms of market. Is it okay, my answer to the
Yes, is it okay? It's fine. But I have another question. I was curious about something. In your presentation, you are saying that you used red grapes. That's correct?
Correct.
Are they better than other types of grapes, or is it the same?
Yes, because the composition of the grape pomace, and here it is very important to note that our samples of grape pomace contain not only seeds and pulp and stems, but also some stalks, and that's why we got lower yield a lower yield. That is the point that I shown in my presentation, and that should be improved in terms of practical studies. This composition in terms of white varieties is quite different. Mainly in terms of soluble carbohydrates concentration. And why have we chosen this red grape pomace? Because was the type of grape that had in that time was being processed in the enterprise, that the local enterprise that provide us the samples.
Okay, thank you for the answer. I have one more before closing the session. This one is for Flora. Flora had 2 works, only one question. The question is regarding your second work. I was curious about why did you use soil samples that came from the Quinta de Gonçalo Martins Guarda? What was the reason?
Because in my master dissertation, I was located this place in the master my master thesis, and after with GIS, and I located this place to collect this soil, and after that, in my PhD, because my supervisor have this region study in I don't know with the aquifer, the depth of aquifer, and the other parameters. And after that, in my PhD, I collected the samples of soil in the same place.
Okay, okay, but the soils have intrinsic properties depending of the location.
Yes, but this soil is a granitic. It's a good soil to infiltration because is a granitic residual soil with low percentage of clay. So it's very good to infiltration. Because soils with a higher percentage of clay difficult and clogging during the time. So this soil and also the fine fraction of the soil have a good properties to remove these pollutants and other pollutants, namely heavy metals and other pollutants that I was studying in my PhD.
Okay, thank you. Thank you. Well, we are about to end. There's no more time for questions. And now, I think all the works were very interesting. I enjoyed it a lot. I would like to thank the authors that were here with me in this session, Professora Flora Silva, Professora Flávia Matias, Professora Isolina Gonçalves. Thank you very much for your contributions and also for your clarifications. It was very interesting. And also, I would like to thank all the viewers that were here with us, not here at home, but participating here with questions. And a final note for this session, we will now have a break of 2 hours for lunch. And the Congress will continue later on at 2:30 p.m. With the technical session number 7. And this technical session is the last one, and the chair will be Professor António Albuquerque. See you again in the afternoon, and goodbye.